investigaciones, sus proyectos de tesis, pero abrimos los 2 días con una presentación plenaria de un investigador de peso. En este caso tenemos 2, hemos tenido la suerte de poder atraer a 2, uno senior, como es John Pannell, para que la mayoría lo conocéis, el otro es un junior, pero de bastante peso en el campo de la ecología evolutiva de plantas, como dijimos Van der Niel. Y invito ya a que suba John Pannell, que nos va a dar una conferencia sobre evolución de los sistemas sexuales en plantas. Y John, cuando quieras. Bueno, en un principio, sabéis, igual que otras veces, yo no sé si os lo pedí, que la mayoría, si podéis, la mayoría lo habéis hecho, la presentación la tenéis en inglés, se puede hablar en español o en inglés indistintamente, pero si os hacen la pregunta en inglés, intentad, porque yo sé que todos, o la mayoría de vosotros, habláis inglés sin problema, pues para interaccionar con nuestros colegas, pues usaremos para dar la charla y seguro que la de directamente en español, en castellano o en inglés.
Right, well, good morning. Thank you very much for inviting me, Fernando. It's a huge pleasure for me to be here. And I know many of you in the audience, and it's great to be back in Spain. I've been in Andalusia for a few years now, and it's a huge joy to be back in these beautiful parts. What I wanted to do today is, in a sense, reflect back on some work that I've been doing for a long time, and in fact, in a sense, winter and spring 2014 marks an interesting anniversary for my research, because it was exactly 20 years ago, this month almost, in January 1994, I came to spend an extended period of time here in Andalusia. I was based at the University of Sevilla to continue doing my doctoral research. And I was working on this ugly little plant here, Mercurialis annua, which many of you will know. It's very common throughout the Iberian Peninsula, particularly common here in the south of Spain and in North Africa. And so it seemed a very nice opportunity to reflect back on some of the work that we've been doing with this plant, but also to illustrate some other work that we've been doing on a couple of other systems over the last few years that develop similar themes. And so I've subtitled my talk A Homage to Males, and you'll see why the emphasis is on males in a moment. But I could also subtitle this A Homage to Mercurialis annua, because it's kept me going for 20 years of research. Several PhD theses have now been done on this plant, several postdocs, and it's kept me very excited about various aspects of biology. And so I started working on this plant with a particular question in mind, and that was what are the implications of the evolution of separate sexes of dioecious? And I was specifically, when I came to do my PhD, I had been working on 2 species of dioecious plants in Australia for my master's, which were dioecious but long-lived perennials, Both of them were serotinous species, maintained their seeds in the canopy, and that will be a theme which we'll come back to a little bit later in my talk. But I wanted, at the end of my master's, I started thinking about the implications of transitions between hermaphroditism and dioecy, and I wanted to find a plant that was easy to cultivate and do experiments with, in other words, an annual plant, if at all possible. And so I settled on Mercurialis annua, which is mainly dioecious through most of its range, as you'll see a little bit later. But down here in southern Spain and North Africa, one finds very interesting polymorphism, which then became the focus of my PhD. But since then, we've continued to use this plant as a model system to address questions about the evolution of sex ratios, sex allocation, the evolutionary genetics and population genetics of metapopulations. We're now doing experimental evolution studies with populations of Mercurialis annua, and we have just finished sequencing the genome of diploid Mercurialis annua in an attempt to understand the evolution of sex determination and sex chromosomes. So you can see how this ugly little plant has become quite an interesting model for my lab, and in a sense, one of the messages that I'd like to bring to you today is that actually this plant is growing on your doorsteps here in Spain, and one way in which I'd like to illustrate this as a potential model for use as biologists is a way in which one can use the plant for practical work with students, for example. So let me just start off with this rather dazzling photograph of flowers and inflorescences. What you see here is a range of hermaphroditic species. Most plants, of course, most flowering plants are hermaphrodites, I don't need to tell you that. But if we think about the history of the study of hermaphroditic plants, and consider how particularly ecologists, plant ecologists, have considered estimating fitness, and you cannot, I do this little test with students of mine. I ask them, how would you estimate the fitness of a particular plant? They typically answer that ultimately you need to count the number of seeds they produce. And I think that's something that slightly naive students quickly pounce upon, but of course if you look through the literature of particularly population dynamics, population ecology, and evolutionary ecology of plants, you find many references to seed production as the currency of fitness. But this is, of course, a very seed-centered view, and it's interesting to reflect on where this comes from. In some sense, it's the obvious thing to do, just to count seeds, but the great and the good of our field have considered the seeds as particularly important as a currency for investigating plant population dynamics and also the evolution of different strategies. And here's a quote from none other than John Harper, who has, in a sense, established the field of plant demography through his many years working in Wales. And here is a quote from his very famous book: "A plant is the only means with which a seed produces more seeds." In a sense, one can also extend this to, or narrow this down to being, an expression about the purpose or the importance of flowers. Flowers are there to produce more seeds. But this is, of course, a very seed-centered view. In a sense, it's a very interesting way to think about plants and the purpose or the function of a plant, that it is simply the way in which a seed produces more seeds. And it's no coincidence, perhaps, that in 1977 coincides with, it was 1 year after Richard Dawkins published his book, The Selfish Gene, in which he focused attention on the purpose of organisms being simply to allow genes to produce more genes. And so perhaps this phrase taken from Harper was a reflection influenced by Dawkins' book. But of course we as evolutionary ecologists of flowers and pollination know that this is a very one-sided view and a misleading view, because what about the pollen? Half the genes in the population are transmitted through pollen if we're dealing with autosomal genes. And of course, if we wanted to understand floral function and the sorts of things that attract us to study biology as plant evolutionary ecologists, we need to consider both seeds and pollen. And of course, there's been a huge amount of work from both a theoretical point of view, but also empirical studies that have attempted to estimate fitness through both the male function and the female function to come up with a better understanding of how flowers have evolved. And I think it's fair to say that many of the adaptations one sees in flowers are the result of selection through the male component of fitness, and perhaps not so much to the female component of fitness. And I'm sure you're very familiar with much of this theory. When it comes to actually introducing undergraduates to these ideas, and I find this is a very exciting way to bring our research that we're doing on often very detailed and esoteric topics to a wider audience, which is our undergraduates. If we can't explain and excite our undergraduates about our work, then I often think, what's the point of what we're doing? Then it's interesting to consider how we can bring this tension between theory and observation and the way students respond to plants and the way researchers, because we're all just students fumbling away trying to understand biodiversity, how we can bring these two different avenues of thinking or of science, theory and data, together. And so I found that a little bit of theory can go a long way in helping to open undergraduates' eyes to seeing flowers in a new light. And so I'm going to describe to you an experiment that I've done with students over the last decade or so. When I was an academic in Oxford, we'd come down to the Iberian Peninsula in spring every year and look at flowers. And there are a couple of exercises that we did to try to show students not necessarily simply to name all the plants, but to consider the function of the plants that they were looking at. And a couple of the exercises were focused specifically on reproductive biology. And so, if we now simply introduce to students this simple idea that flowers are clearly there to attract pollinators, they're insect or animal pollinated plants, and that in some way fitness is going to depend on how attractive plants are to pollinators. This is something that students quickly appreciate. And then we point out that there are two ways in which plants can achieve their fitness. For seed production, they don't need many visits of pollinators whatsoever. Often a single visit will be sufficient. And so increasing attractiveness beyond a certain point is really not going to benefit the plant's female function very more. But it's easy to explain to students that attractiveness can certainly benefit the male component of fitness, because successive visits of pollinators can continue to deliver pollen to other individuals in the population, and therefore increase the potential siring success. So this is, of course, just Bacon's principle. There are lots of reasons why we as evolutionary ecologists might question this, but as a first approximation when entering the field, it's a very nice rule of thumb. And then with this simple idea, we send the students back into the field and ask them to have a look at flowers. We mark out a number of different species, and they have to take a look and consider how this principle is being played out in the field as a result of natural selection. And here's an example. I'll just name one example here of this Scrophularia, which the students discovered had a very interesting way of delivering its pollen, and that is that, as many of you probably know, these little flowers have 4 anthers, but the anthers don't disperse their pollen all at the same time. The anthers are delivered or are opened or extended one at a time over a period of several days. So the 4 anthers come out and they basically are dispensing their pollen over a period of time. Why? Well, it's easy to discuss the possible implications of that in terms of siring success, increasing the paternal fitness in the population. Another thing that is quite exciting to do with students is to take them to an area where there are polymorphisms. They don't tend to look at more than one plant, but if you encourage them to start looking at more than one plant, they start recognizing, and sometimes they don't see it, which is interesting as well. It's a lesson about how poor we sometimes are as being observers. But if it's an obvious polymorphism, they'll notice it, and you can then ask them to start thinking about the significance of this. And so here is an example of two students who were led to a population of Mercurialis annua, amongst other things, and they recognized that there were two plants that looked very similar, but in which the sexual system seemed to be a polymorphism. And so here they discovered a population in which there were males and hermaphrodites, and as I said before, Mercurialis annua is a species that has this polymorphism. Here is a picture which illustrates a male on the right-hand side, a female on the left-hand side, and this little insect here, inset, is a close-up of the monoecious inflorescence one finds in the androdaceous populations here around in Andalusia, where we basically have a pistillate flower, a female flower, surrounded by a cluster of male flowers. And so here, in populations around Puerto Real, you find basically plants that look like this and plants that look like that. A very different inflorescence architecture and differences in sex allocation as well. And so we then asked the students to come up with some sort of hypothesis as to how this particular polymorphism might be maintained. So, having discovered this polymorphism, we take the students back to where we were staying, and after dinner we say, right, we're going to now derive a model to explain or to predict the male frequencies in the populations. Now this is the sort of thing that David Lloyd and Brian and Deborah Charlesworth and others had been doing in the 1970s and 1980s. They're sophisticated, very interesting papers. But actually when you look at the models, the basic essence of these models is very simple and requires extremely simple algebra that students can do. So we simply tell the students to think about what might be going on in a population in which there are males and hermaphrodites, to make assumptions that will allow them to piece together their model, and then come up with an explanation or prediction for the male frequencies. And so, for example, and of course they need some help with this because it takes some experience to know what sorts of things are useful to assume and what things aren't, but by and large we let them lead the way in small groups or in pairs. And so we asked them, or suggest that they might assume that males produce some quantity, M, for example, times more pollen than the hermaphrodites. So males are producing more pollen than the hermaphrodites. That's easy to see when you look at a plant, but it's also something that you can actually measure. And so we can define a parameter M that describes the relative amount of pollen produced by males versus hermaphrodites. Then we say that it suggests that they could assume that basically mating in these populations, it's a wind-pollinated plant, so effectively mating might be random. So pollen grains, whether they're produced by males or females, on an individual basis have an equal chance of siring seeds in the population. That's something one might discuss, but it's a useful starting point, and indeed that's the sort of thing that people have used in the models that have been published. And finally we point out that at equilibrium, once you've come up with your expressions of fitness for males and hermaphrodites, what one simply has to do is assume that if the population is at equilibrium, the fitness, the combined fitness of hermaphrodite through both its male and female function must equal the fitness through male function only of the males. Otherwise the population would not be at equilibrium. So with that equation, one can solve an expression, or derive an expression that predicts the frequency of males in terms of the sex ratio. And with a bit of help, but actually the students with this help end up arriving at this simple expression the sex ratio, the frequency of males, is equal to m minus 2 over 2m minus 2. It's a very simple expression, easily derived by anyone who can do very simple algebra. And then we say, okay, you've made some assumptions. We've got two parameters here that are related in this very simple way. R, the sex ratio, it's very easy to count in the field, and m, the relative amount of pollen, which we can estimate by counting flowers or weighing staminate versus staminate flowers on males versus hermaphrodites. Let's go back to the field tomorrow, and every pair goes to a different population and measures these two, estimates these two different parameters that we need in our model, and then we come back together again in the evening after we've done this exercise and see whether the data actually match the theory. And this is, of course, the way science works. It's what we all do. And this is a simple exercise to bring students' attention to the importance of theory and testing theory when they're actually starting out as undergraduates. So the next day, everyone goes out, does the exercise, and they produce their data. This is the predicted line here, frequency of males plotted against the relative pollen production of males, so that's that little term m. Those are the two values that they predict. So the difference between males and females here is largely in terms of their leaf size, but also the extent to which they flower. And so what we've done is gone into the genus, looked at a number of species, particularly 49 species of leucodendron that differ in the extent to which they're sexually dimorphic, and we measured them for the extent to which, for their branching patterns, stem diameter, leaf surface, plant volume, height diameter ratios, and the years in which they maintain serotinous cones, so that they're maintaining their fruits within the canopy before fire. And we found some interesting results here. This is now our measure of sexual dimorphism in terms of branching, and this is our estimate of the degree of serotiny for species which typically occur in wet environments versus dry environments. And the basic bottom line here is that the more serotinous a species is, the more sexually dimorphic, and that tends to be accentuated in those species that occupy the drier environments, perhaps suggesting that serotiny is expensive. It's particularly expensive in dry environments, and there, where it's expensive, males and females are doing very different things. It's only the females that are looking after their progeny and in the canopy, and therefore they need to adopt a strategy that differs from the males. Here are similar data here plotting the degree of serotiny against soil moisture days, suggesting that the extent to which a plant can afford in this environment to become serotinous depends on how much access those plants have to water. And if you actually look at what's going on with serotinous cones, this is the rate of transpiration from cones of different biomasses in one of the species, Leucodendron linifolium, and as the cone biomass goes up, the transpiration rate increases. These cones are kept alive, they're losing water, and water is a very precious resource during the Mediterranean summers, and you can expect here strong selection on females to optimize their water use efficiency through physiological and morphological attributes. And so what we have here is sexual dimorphism in leucodendron, which is perhaps the result of a balance between, on the one hand, selection on females for their water use efficiencies, and on males to increase the size of their inflorescences in a male-male competitive game. Okay, very quickly now, knowing that there's a red card that Fernando perhaps has got down there. I just wanted to end with one little story here, with another plant that we've worked on here in southern Spain. I worked on this with Fernando actually some time ago. And the basic idea here is, or the problem here, we've got is that it's the plant's called Filarea angustifolia. You'll probably all know it. And it tends to have, it has males and hermaphrodites in its population. And it's been an ongoing puzzle as to how these hermaphrodites are being maintained. What we find puzzling is that the male frequencies are often greater than 50%, and that is just not possible to explain using any sort of model. And so Fernando and I thought that maybe it was the result of the way we were counting sex ratios, and so we looked at sex ratios in south-facing versus north-facing slopes in southern Spain and Portugal, and what we find here is that the male frequencies were quite low actually in these populations. These were the flowering males at about 30%, and they did not differ between the north-facing and the south-facing slopes, but the proportion of the females, or the hermaphrodites, changed dramatically. There were far fewer in the flower on the the south-facing, dry, hot south-facing slopes than on the less stressed north-facing slopes. And on those slopes we find non-flowering plants. On the stressed slopes you find a greater frequency of non-flowering plants. So that seems to be a nice answer to the question and problem solved. But actually, that was, I think, part of the story, obviously, because I don't think those data are wrong. But there's been some fantastic work being done by some people in Montpellier and Lille, some French researchers, which have been working on the system since the early 90s. And I just wanted to end by highlighting their work because I think it's a spectacular. They've come up with some extremely exciting results over the last couple of years, which were completely unexpected. And I don't have time to go through the details, but you should read the paper. It was published in Science a couple of years ago. And what they find, actually, is that there are two classes of hermaphrodites. The hermaphrodites are self-incompatible, and there's a lesson here because everyone believed they were self-compatible as a result of crossing experiments, but that was all contamination. These are self-incompatible plants. And it's a single locus diallelic system, which is very unusual. It's basically the sort of thing that you would find in primula. It's basically a distyler system without the distyling. And so there are only two different hermaphroditic morphs. Those hermaphrodites are self-incompatible, and they can't mate with half the population of hermaphrodites. They can only mate with the other half. So hermaphrodites immediately have a siring disadvantage. But males have invaded the population by being able to mate with all hermaphrodites. So they immediately have this big advantage in siring, simply because there's a female sterility mutation that's evidently linked or somehow co-functional with the self-incompatibility locus. And that's just amazing. And so basically, and what's particularly interesting is the phylogenetic context here. This is a large family, the Oleaceae, where Filarea is down here. Back up here we have jasmine, and jasmine is distylus. So is it possible that distyly evolved here and has been retained in all of these species in a non-distylus form, but the self-incompatibility system has been maintained? And you find in many of these species, particularly in the genus Fraxinus, males popping up again and again. Androdaceae is really common in this family, whereas it's almost absent throughout the rest of angiosperms. So here we see something that, from a phylogenetic point of view, would be extremely exciting to trace through the family the self-incompatibility system. And we're busy collaborating now with Miguel Berbu in Valencia and people in France to try to figure out what's going on in Fraxinus. So that's something to talk about in future years. So I just wanted to end once again by thanking Fernando for inviting me and all these people who've helped with these projects over the years, and thank you for your attention.
Thank you. I think there is time for a few questions. If you want to...
Tengo un micrófono aquí.
Pero, ¿se oye bien? Yo creo que lo mismo, si se puede sin usar el micrófono y hablamos un poco, yo creo que es mejor que ir con paseando. Bueno, ¿alguna pregunta?
Hi, John. In the phylogeny of Oliaceae, there are lots of shifts. You can see. Can you please explain? Yeah. What's your hypothesis of so many shifts in a big family like that in the course of evolution? I mean, do you have any general idea why it's so frequent here in this family and it's not so much in some other families?
Yeah, so the question is, firstly I should point out that this is the phylogeny that was done by Eva Wallander in Sweden for her PhD thesis, so I'm just sort of stealing her phylogeny here. But the question is, why do we find within this family so many of these fascinating shifts between combined and separate sexes? And the answer is no, I don't. I think it's clearly something to do with the family, obviously, and I wonder whether the discovery made in France by Pierre Saint-Laprade and his family and his colleagues of what's going on in Filaria and Angustifolia, whether that may be providing us a clue, because it's a very unusual trait to have a distylus, biallelic self-incompatibility with without the morphological associations. And so if that's what's going on in the background, we've got this very special type of self-incompatibility that might then offer us the species that basically have retained that possibility of evolving these strange systems. And I might just sort of foreshadow a paper that it will be coming out fairly soon, because I saw the manuscript a couple of weeks ago, and it's even more exciting than the science paper. And that is that the same, as a result of the further generation of crosses, the French team have discovered in Filaria that not only do we have this interesting link between self-incompatibility and female sterility, but I think they've discovered the first clear meiotic driver influencing sex ratios in the plant. So not only does this one locus influence sex and self-incompatibility, but there is also a meiotic driver there, and it operates in a very interesting way. That when you make a male with one type of hermaphrodite, produces the one-to-one sex ratio that you'd expect, but when you make the male with the other type of hermaphrodite, the other self-incompatible class, all the hermaphrodites, all of the progeny are male. And it's, I think, clearly this very interesting link between sex determination and self-incompatibility seems to open the way to these strange things happening. So perhaps when we look down this phylogeny, many of these transitions might involve other interesting conflicts between male and female function that seem to be happening in Filaria. So it's going to be interesting to see to what extent we could actually, the French team will be able to work out the mechanisms of what's going on. It's totally mysterious as to how this could actually be achieved at a mechanistic level, but there's almost certainly a fascinating arms race going on at the level of the genome. So if that's the background in one species, perhaps there's all sorts of conflict going on throughout the family.
Juan, yo. Cuando ha hecho Pablo la pregunta, ¿hay trabajos enterales o no?
Sí.
Bueno, me doy la vuelta. How is the self-incompatibility in the rest of the family?
Well, it's not known. And in fact, there's another salutary story here, because several years ago we had a grant funded by the EU to look at populations of Frexes, and as part of that work we actually did look at the mating system, and our conclusion back in the early 2000s, which was consistent with several other people who looked at this in Fraxinus olimus, was that the species was self-compatible, We found evidence for inbreeding depression. We published 2 papers on that, one in the Proceedings of the Royal Society and one in Evolution, about self-compatibility and interesting paternal effects of mating.
But
It's self-incompatible, we got it wrong. And all of the results, we went back and checked using microsatellites 2 years ago.
there is self-incompatibility in other species, even diabetic, in jasmine and forsythia.
Yeah. Yeah, exactly.
So
So,
I mean, how many alleles are in other species self-incompatible in the family? You know?
I don't know, but I wouldn't be surprised that if perhaps in all of these systems, maybe we'll find diallelic self-incompatibility. Yeah. But we don't know. We still have to do the work. And I guess the point has been that quite a few people who worked very carefully produced results that looked as if the species was self-compatible with all contamination. And the lesson is, if you're working with wind-pollinated plants, you've got to check with microsatellites. Because
by the way, there is another case of linkage between self-incompatibility and sex determination, erythropsis. But the result is the opposite. It's a genotypes species.
okay. Yeah.
But this is, I think, it's by chance. This is the only case in erythropsis.
Okay.
It's only just one species.
Tiempo para una pregunta rápida. Desde aquí no se ve muy bien. Cuando tengáis alguna pregunta, levantad bien la mano. Bien, pues muchísimas gracias por la charla. Tenemos ahora un descanso antes de empezar con las comunicaciones. Recordad, el ambigú está en la primera planta y podéis tomar café. Tenemos media hora para el café. ¿Se debe puntual?
Bueno, sí, están acostumbrados.
Sobre la